Namaste. I am JV'n. I am Dr. Anupama Goyal. I am working as Associate Professor in Botany in Jyoti Vidyapith, Women's University, Jaipur. Today I will deliver my video lecture on stellar system and heterospore in pteridophytes, and this lecture is useful for BSc as well as for MSc students of all the private as well as government universities. So first of all, I talk about the stellar system of pteridophytes. Well, what is a stele? The conducting system of pteridophytes consists of xylem and phloem and is associated with parenchymatous cells, all of which are organized into a stele. The term stele has been derived from a Greek word meaning pillar or column. The concept of the stele as the fundamental unit of vascular system was put forward by Ventigum and Doliet in 1886, who proposed and developed the stellar theory. So the stellar theory was developed and proposed by Ventigum and Doliet, and stele is the fundamental unit of the vascular system. Now, according to the stellar system, according to the stellar theory, the primary structure of the stem and root were fundamentally similar in gross anatomy because both consisted of a central core, the stele, surrounded by the cortex. Now, the term stele was interpreted as the vascular tissue and conjunctive tissues associated with them, and the pith and pericycle if present. The stele of stem was connected with that of the leaf by a vascular connection known as leaf trace. Now I come to the point, what are the types of steles which are found in pteridophytes. On the basis of the kind of stellar organization present in different pteridophytes and evolutionary sequence can be recognized among the different groups. The stele in pteridophytes can be differentiated into two major groups. First is the protostele. Now, what is protostele? So, it is the most simplest and primitive type of stele. In protostele, the vascular bundle is a concentric solid mass and the central core of xylem surrounded by a layer of phloem, and finally it is surrounded by a layer of pericycle. So Jeffrey in 1898 named primitive type of stele is the protostele. So protostele is the most primitive type of stele found in pteridophytes, and in this there is a solid central mass of xylem surrounded by phloem, and there is no pith. It is considered to be the most primitive because it is present in some earliest known land plants that appeared about 400 million years ago. The protostele exists in the following forms. So first of all, see the diagram showing the haplostele and actinostele, the types of protostele. Now I show you this A diagram shows the haplostele and this B diagram shows the actinostele. So now I talk about the types of steles in pteridophytes or haplostele. First of all, haplosteel, then actinosteel. So what is haplosteel? It is the most primitive type of steel. In haplosteel, the xylem forms a solid mass and it is smooth and spherical central core which is surrounded by a continuous layer of phloem. For example, haplosteel can be seen in Lygodium and Selaginella. This haplosteel has been regarded as the most primitive among the different types. Now, what is actinosteel? This is the modification of the haplosteel and it is somewhat more advanced in having the central xylem core with radiating ribs, and phloem is not concentric but the phloem is present in between the radiating ribs of the xylem. So actinosteel can be seen in xylotum, which is a pteridophyte, and the protoxylem is present at the tips of the radiating arms. So here I show you again the picture or the diagram. See, in this actinosteel, or what I told you just now, actinosteel, see the B picture. In this, the xylem is in the form of radiating ribs. These are the radiating ribs of xylem. Matlab, xylem is present in the form of radiating ribs and phloem is not concentric and it is present in between the radiating ribs of xylem. So this is the phloem which you see in between the radiating ribs of xylem. Now next slide is of plateausteel. So what is plateausteel? In the stem of some species of Lycopodium, the solid core of xylem gets broken in a number of plate-like lobes, more or less lying parallel to one another. The phloem alternates with the xylem plates. For example, Lycopodium voluvile. This specialized form of protosteel is called as plateausteel. Now, what is mixed protosteel? In this type of mixed protosteel, masses of xylem and phloem are uniformly distributed. Scattered groups of xylem are embedded in the ground tissue of phloem, as for example in Lycopodium cernum. Now I talk about the protosteel with mixed pith. In the center there is parenchyma cells associated along with the tracheids. For example, lepidodendron. Lepidodendron is one of the pteridophytes in which protosteel with mixed pith is found. It is derived from the transformation of the tracheids into parenchyma and may be first step in the formation of the pith. Such type of protosteel is called as mixed protosteel. So here I show you the diagram of the plateausteel and mixed protosteel. So look at this diagram of plateausteel. In this the xylem is broken into a number of plate-like lobes. These lobes, the xylem is broken into a number of plate-like lobes, and these lobes are arranged parallel to each other. So it is called as plateausteel. And in this diagram you are seeing the mixed protosteel. So in this mixed protosteel, masses of xylem and phloem are uniformly distributed. You can see xylem and phloem, they are uniformly distributed in this diagram. Now I talk about the radial protostele. This stele consists of radial vascular bundles enclosed by an endodermis layer. This type of radial protostele is found in root and stem of young stage of some species of Lycopodium. Now, what is siphonostele? This is a kind of stele in which there is pith. There is pith in the central region, and the presence of pith in the center says or shows that it is an example of siphonostele. So siphonostele is a modified form of the protostele which has developed central pith, so it is called as medulated protostele. So siphonostele is called as medulated protostele. Now the question arises, why? Because there is the development of the central pith, so it is called as medulated protostele. And this type xylem is in the form of a hollow cylinder enclosing a well-defined central pith in the center, and xylem is surrounded by concentric phloem. This type of stele is thought to have been evolved from a protostele by the conversion of tertiary elements into parenchyma. Now I talk about the origin of pith in siphonostele. Well, students, just now I told you that there is the development of pith in the center, so it is called as siphonostele. Now the question arises about the origin of siphonostele. There is a general acceptance that siphonostele is evolved from a protostele. So two theories have been proposed to explain the origin of pith. First is the intrastellar origin of pith. According to this view, pith has originated as a result of the transformation of tertiary elements of the central xylem core into parenchyma. Thus, the pith is totally intrastellar. This view, that is intrastellar origin of pith, has been supported by Boodle in 1901 and Vaughan in 1908, and it is found in Osmunda regalis, Botricium, Turnware, and tracheids are scattered throughout the pith. Now I tell you about the extrastellar origin of pith. This hypothesis was supported by Jeffrey in 1902 and 1910. According to the view of Jeffrey, protostele has transformed into siphonostele due to the migration of cortical cells into the stellar axis openings such as leaf and branch gaps probably provided passage for the invasion of parenchyma. This view derives support from amphiploic siphonostele that has two endodermal layers. One delimits the stele from the cortex and the other delimits it from the pith. Since endodermis is a structure peculiar to cortex, the pith is considered as cortical in origin. So now I talk about the types of siphonous steel which are found in pteridophytes. Now, according to the distributional patterns of xylem and phloem, the siphonous steel is classified into the following two types: ectofloic siphonous steel and amphifloic siphonous steel. So, what is ectofloic siphonous steel? In this type, the central pith is surrounded by concentric cylinders of xylem and phloem. One of the best example is equisetum. The phloem is present only on the outer side of xylem. So why it is called as ectofloic siphonous steel? Because phloem is present on the outer side of the xylem. And now I talk about the amphifloic siphonous steel. So in this type, there is pith in the center. The concentric xylem is surrounded on both the sides, that is externally and internally, with phloem cylinder, which is followed by pericycle. Means in amphifloic siphonous steel, the xylem is surrounded on both the sides with the phloem cylinder. In its simplest form, the siphonous steel has no leaf gaps. Leaf gaps are not present in siphonous steel. This type of siphonous steel is termed as cladosiphonic siphonous steel. So, in cladosiphonic siphonous steel, leaf gaps are absent. For example, some species of Selaginella have cladosiphonic siphonous steel. On the other hand, a siphonous steel with leaf gaps is termed as phyllosiphonic siphonous steel, which is found in Marsilia. Now, I talk about first I show you the diagram of the ectofloic siphonous steel and amphifloic siphonous steel. So look, a diagram shows clearly the picture of ectofloic siphonous steel, in which phloem is present only on the outer side. And in B diagram, which is the diagram of amphifloic siphonous steel, xylem is surrounded by the inner phloem as well as by the outer phloem. So this is the siphonous steel. Now I talk about the U steel and solenoid steel. So what is U steel? Here, the hollow vascular cylinder with central pith gets broken into a number of collateral bundles arranged in a ring. This type is also known as polyfascicular siphonous steel, which can be seen in equisetum. Now, what is solenoid steel? This is modified form of siphonous steel which gets perforated by a single leaf gap. The solenoid steel may be either ectofloic or it may be amphifloic. This solenoid steel is found in ferns. Now, what is dictio steel? So, a dissected siphonous steel is known as dictio steel. In this type, the cylindrical steel is interrupted by numerous leaf gaps that overlap each other. In the cross section, the stele appeared as discrete strands or bundles, each with internal xylem and concentric phloem, and is called as meristele. So pteris is the best example of. Now I talk about the polycyclic stele. So now what is polycyclic stele? This type of stele is most complex one among all the pteridophytes. A typical polycyclic stele possesses two or more concentric rings of vascular tissue in regular rings or cylinders, one within another. This may be a solenoidstele or a dictyostele. Now, this may be a solenoidstele or dictyostele. Two concentric rings of vascular tissues are found in pteridium, aculinum, and the three concentric rings are seen in metonia pectinata. Now I talk about the evolution of the stellar system, how the stellar system evolved in pteridophytes. Well, it is generally held that the simplest type of stele is protostele. It is fundamental type for the vascular plants in general and the pteridophyta in general, and all the other types of stele have been derived from it in the course of evolutionary specialization. As far as is known, all pteridophytes in the initial stage, in the sporulation stage, starts with protostelic stem. It is permanently retained in the adult stem of many living pteridophytes, for example, Selaginella and Lycopodium. A protostele may serve the need of small shoots and also of the larger stems under special circumstances, but where the shoot grows accompanying an increase in size, there is internal differentiation of the stele. Now I show you, this is the slide of the evolutionary stele. Now I talk about the heterospory. So heterospory is defined as the presence of two types of spores in the same plant. So two types of spores are present in the same plant and it is called as heterospory. The two kinds of spores are different in shape, size, structure and function. One spore is small and the other type of spore is larger in size. So the smaller spores are known as microspores and the larger spores are called as megaspores. The microspore is produced in microsporangia in large number and megaspore is produced in megasporangia in a small number. So now the question arises, what is the small spore? A small spore in pteridophytes is called as microspore, and what is the large spore? Large spore is called as megaspore. And now this microspore is formed inside the microsporangium and megaspore is formed inside the megasporangium. Now this differentiation of spores in size is definitely related to the distinction of sex of the gametophyte. The smaller spore or microspore on germination leads to male gametophyte, means microspore on germination gives rise to male gametophyte, and the larger spore called as megaspore on germination gives rise to female gametophyte. Among the pteridophytes, heterospory occurs in 9 genera. So what are the 9 genera in pteridophytes in which heterospory is present? They are Flagella, Isoetes, Stylites, Marsilia. Marsilia is in the course of BSc and MSc. In this also heterospory can be seen, and heterospory is also found in Pilularia and in Salvinia, in Regnelidium, Azolla and Platysoma. Now I talk about the origin of heterospory in pteridophytes. Well, a detailed study of homosporous forms has revealed that heterospory originated due to the reduction in number of spores within sporangia, means there is reduction in the number of spores in the sporangia, and so the heterospory originated. That is, if more number of spores are functional, then there is more competition for nutrition and limited nutrients are provided to developing spores. In this way, microspores are formed. They are smaller in size and they are more in number. On the other hand, if some of the spore mother cells in a sporangium disintegrate during development, the remaining ones get sufficient nutrients. These are the megaspores. They are larger in size but less in number. Now I talk about the evidences of heterospory. So first of all, there is one important evidence for heterospory in pteridophytes, that is the paleobotanical evidence. These were concluded on the basis that earlier vascular plants were heterosporous. For example, Lepidocarpon, Lepidocarpon and Lepidostrobus, they were heterosporous. So, according to Scott in 1894, an indication of heterospory can be traced in Calamostrachus binianda, Calamostrachus species and Calamostrachus casciana. In Calamostrachus biniana, the sporangia were with large number of small spores in tetrads, whereas in Calamostrachus casciana, two distinct types of sporangia, microsporangia as well as megasporangia, occurred. What I mean to say is, in Calamostrachus casciana, two types of sporangia were found, macrosporangia as well as microsporangia, whereas in Calamostrachus biniana, only one type of sporangia were found with